Hello everyone, I am Dr. Bibu Prasad Pandas, Senior Scientist Formulation Research Development, welcoming you all to the second part of the lecture Preformulation Studies, Formulation and Pharmaceutical Science Perspective. This part of the lecture is sponsored by Galt Pharmacy under the philanthropic vision of quality pharmacy education for all. So the we are discussing about the different part of this pre-formulation stages. Under the bulk characterization, we are talking about the molecule or drug substance, how it is going to be characterized. Under that last lecture, we talk about the details about the characterization of the solid compounds. Under this part is called polymorphism. Polymorphism, when a substance exists in more than one crystalline form, the various forms are called to be polymorphs, and the phenomena is called polymorphism. Polymorphs is solid material with two or more molecular arrangement which give a distinct unit cell. Crystalline polymers have same chemical composition but different internal crystal structure. Therefore, they possess different physicochemical properties. So polymorphism, as we understood that the most of the crystalline compounds having exist more than one forms, and that process make the solid compound into a polymorphism and polymorphs. And this polymorphism is very common in pharmaceutical drugs, and it is quite difficult and challenging to predict. Most of the organic and inorganic compounds of pharmaceutical relevance can exist more than one or more crystalline form. The common crystalline forms found in a given drug substance are called to be polymorphs or solvents. Solvents. In general, the more stable polymorphs will have a higher melting point and lower solubility. So this is the reason we have to understand why polymorphism is play a important role in a formulation development, because most of the polymorphs have a different physicochemical characteristics. Those physicochemical characteristics makes a challenge in understanding this molecule or drug substance, because most of the time you will find difficulties in understanding this molecule in terms of their solubility or in terms of the bioavailability or in terms of the physicochemical properties to see that how it's going to understand this molecule's behavior in formulation development and going to play a very critical role in making up a suitable dosage form or suitable formulation development of a for a API. The lesser stable polymorphs have chances to get convert into a stable morph over the time during due course of the state of the storage conditions. We have noticed that most of the stable polymorphs get converted into a more stable, and by the way the physicochemical properties of the molecule get changed. So under this polymorphism, during the process of crystallization, some compound have tendency to trap the solvent molecule within the structure. So that we call molecular adducts. And these molecular adducts are different types. One we call non-stoichiometric inclusion compounds or adducts. In this crystal, we will notice that certain solvent molecules are get entrapped within the crystal lattice, and but the number of the solvent molecules are not included in the stoichiometric number. That's the reason we call non-stoichiometric inclusion or adducts. So depending on the shape, there are three types. That the first one is the channel, when the crystal contain the channel in which the solvent molecule can be included. Example is urea forms a channel. Second type of non-stoichiometric inclusion compound is the layers. Here the solvent molecules are entrapped in between the layers of the crystals and the clathrates or the cage. In this process, the solvent molecule are entrapped within the cavity of the crystal forms of the all sides. The second type of the molecular adduct is called stoichiometric inclusion compounds or stoichiometric adducts. This molecular complex has incorporated the crystalline crystallizing solvent molecule into a specific site within the crystal lattice and has a stoichiometric number of the solvent molecules are complex. So in this stoichiometric inclusion compound, here the solvent molecule entrapped into the internal structure will be going to be accounted in the stoichiometric numbers. So when the incorporated solvent is water, then we call it as a hydrate. When the solvent other than the water, we call it as solvents. Depending on the ratio of water molecules within the complex, now it can be nomenclature H anhydrous when 1 mole of the compound with no mole or no molecule of water. Similarly, hemihydrate, half mole of water, monohydrate, 1 mole compound with 1 mole of water, dihydrate, 1 mole compound with 2 mole of water, inclusion into the stoichiometric adducts of the compounds. Polymorphism is very common in a pharmaceutical formulation development or pre-formulation study. You will find many of the drugs or excipients are behave these polymorphic behaviors or polymorphism characteristics. Active pharmaceutical ingredients, mostly the drug, example is chlorphenical palmitate, is the most classical example normally we cite to understand the polymorphism in the pharmacy or in pharmaceutical product development. Ritonavir, cortisone acetate, carbamazepine, and some pharmaceutical example under this category of the excipient. Again, the cocoa butter we also consider as a classical example in formulation development of how the excipient going to influence in the formulation development, and the polymorphism and formulation development is going to be a most challenging part in making of a suitable formulation development of a API. Nearly all long chain organic compound exhibit a polymorphism. Fatty acids different attachment of carboxylic groups to adjacent molecule which modify the angle to tilt the chain, and that causes the polymorphic behavior of the compound or the excipients. Few example, as I said, the classical example is the chlorphenical palmitate, which is a ester prodrug of chlorophenical, which is an antibacterial agent, and it is noticed that this chlorophenical palmitate having a three different crystalline structure or three different polymorphs. The polymorph A, which is considered to be stable but biologically inactive because it dissolves too slowly to get absorbed and reduce the bioavailability because of slow solubility or slow biological absorption to the systemic circulation. Whereas the in polymorphic forms B, which is considered to be a metastable, but it is a biologically very active. Similarly, another form called polymorphic form C, which is totally unstable. So, looking into this characterization of the chlorophenical palmitate, the polymorphic polymorphs and polymorphism behavior, the United States Pharmacopoeia has placed limit of 10% use of polymorphic form A in the oral suspension dosage form development. So formation of form A can be happen. So there is chances of conversion of B to A or C to B or C to A may possibly happen during the process of storage, during the process of grinding, or during the process of milling at high temperature. This changes forms polymorphic changes of the one compound from one polymorphic form to another polymorphic form. So it is noticed that polymorphic form B and C get converted in during storage conditions and to a polymorphic form A. So this is a classical example to understand that polymorphic behavior. How does it influence the overall formulation and the bioavailability of the drug? To understand this experiment indicate that how the polymorphic form A, you can see that this polymorphic form A having a low bioavailability or plasma drug concentration or serum drug concentration is lower in the A, the polymorphic form, whereas the B, A and B forms having 50-50 polymorphic combination having a the plasma better plasma drug profile or serum drug level of chlorphenicol palmitate compared to A, whereas the B is having a much better compared to A and A is to be 50-50. It indicates that these polymorphic forms having a different role and different way of these physicochemical properties changes will have a greater influence in the bioavailability and the absorption of the drug molecule to the systemic circulation. So another example is ritonavir. This molecule was marketed in 1996, and during this process of the development, it is noticed that most of the product, most of the formulation of this molecule of this ritonavir molecule failed the dissolution test, and it is noticed that because of this crystalline nature of the compound, it is didn't reduce the solubility and it failed the dissolution study. So later stage they have studied and they found that these polymorphs have a different forms. Form 1 called trans conformation and form 2 is a cis conformation. So there is a two polymorphs are identified and these two polymorphs have a different physicochemical properties. Looking into this retinovirus polymorphs, we have noticed that these polymorphs having a different conformational arrangement. So this polymorph A, cis and trans having different crystal habits. So this difference in crystal habits leads to different physicochemical properties of the molecule, and it is noticed that because of this the polymorphic form too have a poor solubility, and because of that you will find the solubility, dissolution and bioavailability of these two polymorph form 1 and 2 have a distinct characteristic differences in their physicochemical properties and overall the performance of biological performance or bioavailability of the two morphs. As I said, most of the API or the drug and also the excipients which are therapeutically not to be active in making of the formulation, but these excipients also have the polymorphic behaviors. And because of this polymorphic behavior of the excipient, whenever we are using the formulation development of any pharmaceutical ingredient or active pharmaceutical ingredient into a dosage form, this polymorphic changes or behavior of the excipient also greatly influence the formulation development, even overall bioavailability and the of their formulation or of the API. So the classical example is cocoa butter. As you know, the Theobroma coca seeds normally used to get the cocoa butter, and mostly these cocoa butters are used in the pharmacy formulation of suppository base, ointment, creams, etc. So it is noticed that the cocoa butters having 34 to 36 degrees centigrade melting points and it exhibit more than six polymorphic forms, and these polymorphic forms having six polymorphic forms having a different physicochemical properties starting from the melting point to their behavioral changes. So it is characterized, it is analyzed, and it is clearly distinctly noticed that these different morphs have different melting point and their different physicochemical properties. This slide depicts that this uh, polymorphic forms of the cocoa butters as analyzed, and the each polymorphs have identified a different melting point by doing DAC study, difference scanning calorimetric study. So the polymorphism is going to play a very important role in the formulation development of a stable dosage form or stable API with a stable dosage form. So That's the reason we have to characterize the polymorphic behavior of the drug substance along with the excipient to get a stable and a reliable and understandable and stable dosage form so that which can be take our molecule into the longer term to the bedside to the patient to can use this molecule and dosage form for the therapeutic application. So transformation between polymorphic form can lead to a formulation problems. As I said, most of the time these polymorphic behaviors get changed. The polymorphic forms get changed due course of the time. Like keep on storing for a long period of time, the metastable convert into to non-stable and unstable or non-stable forms will convert into the metastable. These polymorphic changes do observe in different compounds. So that changes may causes a critical and challenges for the formulation scientists to understand how this molecule going to develop in due course of the time. So this is called phase transformation. The molecule get transformed from one form to another form. This is called phase transformation of polymorphic behavior of the drug or API or excipient. Due to this polymorphic transformation, normally we have noticed that there is a change in the crystal size, and the crystal size does play a very important role in a suspension formulations or a emulsion characterization. And the most important part is in case of the suspension development, we have noticed that sedimentation and caking formation of the suspension has notice during the change of the phase transformation of the polymorphs. And due to that, you have also noticed that the creaming of the grittiness of the formulations or suspension or emulsions also has noticed due to change of the particle size due to the phase transformation of the polymorphs. Coming to the most important part is even the due to this change in the crystal size, even sometime the alteration in the aqueous solubility. And as we know that if the solubility is altered, there will be alteration in the bioavailability and also lead to the poor bioavailability and the absorption. And then finally, you will find the poorly soluble compounds having this type of challenges, which causes the phase transformation of the molecule. And later stage, we have noticed that these molecules have produced a rate limiting state in drug reaching to the bioavailability or reaching to the systemic circulation or the poor bioavailability. Similarly, we have also noticed that the cocoa butters having a different melting points, and when we are making these suppositories and with the different melting points, the dosage form of suppository deforms happens because of melting points. So the proper selection of polymorphic forms is always be a big challenge for formulator scientists to understand how does it going to influence the making of a stable dosage form has to be characterized. So also we have earlier discussed that the crystals also comes considered to be a stoichiometric, non-stoichiometric. Under that, we have also discussed about the solvent and hydrate. And just for understanding that the solvent and hydrates are also called pseudopolymorphs. So to make you understand what is meaning of pseudopolymorphs, the pseudopolymorphs are nothing but the crystalline compounds which contain some solvent molecules within the lattice of crystal lattice, then it is called pseudopolymorphs. When the when the crystal lattice contain the solvent, we call solvent. Then we call solvents. When it contain water molecule, we call it is hydrates. Crystals without trap of any solvent, we call anhydrates. Many pharmaceutical solids are often synthesized by standard organic chemical method, purified, then crystallized during the process of development. And during this process of development of the compound and crystallization, we have noticed that certain residue of solvent molecules are entrapped into the crystalline lattice, and that lead to the pseudopolymorphs. And mostly, these pseudopolymorphs or the solvents and hydrates have different physicochemical properties, and these different physicochemical properties lead to a challenges or understanding to a develop a stable dosage form. So, like as I said, the solvents and hydrates have a different melting points, aqueous solubility, and most of the time we have noticed that solvent or hydrates have this type of the behavior, this physicochemical property change in the physicochemical properties will be has to be analyzed and properly characterized to make a stable dosage form. Like most of the uh, anhydrous compounds forms a higher aqueous solubility than a hydrate forms. So solvents of non-aqueous solvent have higher aqueous solubility than the non-solvents. Here the example, here we have noticed that example of oxyphenobutazone is a compound. Oxyphenobutazones contain three or four different forms, so pseudopolymorph forms like anhydrates or non-solvent, solvates or solvate with cyclohexane, solvate with benzene and monohydrate forms. So different these solvents have a different intrinsic dissolution rate and solubility. So that's how we have to understand. We have to understand in the process of development of crystallization, as I said, certain organic solvent or solvate will be retained within the in the lattice crystal lattice, and that going to alter our solubility of the drug compound and ultimately the dissolution rate and ultimately going to the bioavailability of the oxyphenobutadiene. So anhydrous forms having a higher dissolution rate compared to a solvent and compared to monohydrates. Thank you very much. And if you have any queries, please put your questions, discussions into the comment sections, and we welcome you all to give your suggestion, comments, all regarding the lecture. If you like, please give a like or share and subscribe to our channel so that we will be take further to next level, and we will try our best to give the all the basic information whatever the required for a academic point of view and industrial point of view. Thank you very much, and see you in the next lecture. Thank you. Stay tuned for the part 3 pre-formulation study. Thank you.